Welcome to the deep dive. Today, we're taking on what is arguably the most persistent problem facing modern artificial intelligence.
Mm.
The fact that it learns brilliantly, but then it forgets catastrophically.
You give it a new skill and boom,
The old skills are just gone.
gone.
It's a huge limitation, and frankly, it's a bottleneck. For all the incredible breakthroughs we're seeing in, large language models and computer vision, all built on deep learning, these artificial neural networks, or ANNs, they just do not have a true lifelong learning capability.
They're just not built for it.
Not at all. They're resource hogs. They need these massive static training data sets, and they are fundamentally incapable of efficiently integrating new information without overwriting the old. It's brittle.
Okay, so let's unpack this. We've been sifting through the research on neurobiology and AI modeling, and what the source material is suggesting is that this failure isn't about network size. It's about missing fundamental biological design principles.
And we're going to look at two big ones today: neuronal diversity and cell-specific neuromodulation.
And if we can model these features effectively, especially inside spike-based neural networks, which are already, much closer to the brain's architecture,
Right.
we can move from these brittle, isolated systems to AI that's adaptive, resilient, and way more resource efficient. It's about building smarter, not just bigger.
That's the mission for this deep dive.
Let's do it.
Okay, so let's start by defining the battlefield here. On one side, you've got the AI status quo, which is isolated learning. This is training a model on a massive bounded data set like, telling cats from dogs. And once it's done, that's its job. It's task-specific, it's often static, and it is computationally exhausting.
And on the other side, you have the biological gold standard, lifelong learning. I mean, think about a human or any complex organism. We're learning constantly.
All the time.
We're integrating new data, a new face, a new route to work, a new language, all while keeping that foundation of prior knowledge.
So the main failure point for AI is when you introduce a new task, the model's synaptic weights adjust so drastically that they just erase the connections for the older tasks.
That is catastrophic forgetting in a nutshell.
And the brain just doesn't do that. So when we look at the source material, what are the core capabilities that let biological systems succeed where AI is failing so badly?
There are really three critical intertwined capabilities. The first one is simply the ability to learn continuously.
Okay.
Biological systems do this because they operate on multiple timescales. I mean, we have sleep and wake cycles that consolidate memory, but down at the cellular level, we have these chemical messengers, the neuromodulators, that mediate something called metaplasticity.
Wait, metaplasticity, so that's like learning how to learn, isn't it?
Exactly, you've got it. It's the dynamic regulation of synaptic plasticity itself. It's not just about whether a synapse gets stronger or weaker.
It's about whether it's even allowed to change.
Precisely. And by how much. This ability to, dial up or dial down the sensitivity of the learning rules, that's how the brain selectively preserves essential connections while allowing new ones to form.
Okay, so continuous regulated learning is number one. But what about the sheer efficiency of biological learning? We don't need a million pictures to recognize a new kind of coffee mug.
And that leads right to the second capability, adaptive knowledge transfer, or what some people call few-shot learning.
Right.
Organisms adapt so quickly, sometimes from just a handful of trials. And this speed, this efficiency, it's primarily managed by those same neuromodulatory nuclei: dopamine, serotonin, acetylcholine, noradrenaline.
The chemical messengers again?
They're context-aware signals that dynamically reconfigure the network's behavior for whatever task is at hand. It allows for this rapid, really high-impact refinement of knowledge.
And the third capability is what makes the whole system actually survive in the messy, real world.
Tolerance to noise.
Yeah.
I mean, if you've ever tried to train a simple AI in a noisy environment, you know how quickly it just breaks. The biological brain is inherently robust.
Why?
Well, because of two factors we keep coming back to. The neuromodulators, specifically noradrenaline and acetylcholine, which dynamically tune the signal-to-noise ratio in specific circuits.
And the second one?
The underlying neural cell type diversity.
Yeah.
A network with different kinds of neurons is just fundamentally harder to disrupt.
This idea of redundancy and flexibility, it points to a concept from the sources I find really fascinating: degeneracy. When you first hear that word, it sounds negative, like failure. But in physics and in biology, it's actually a design virtue.
It is the ultimate resilience mechanism. In statistical physics, degeneracy just means that different microscopic setups can lead to the exact same macroscopic outcome.
Okay, give me an example.
Think about a heating system. You could have 5 pipes running at half capacity, or maybe 2 pipes running at full blast. The room temperature might stay exactly the same.
So the function is stable, even though the internal state is different.
Exactly. Functional flexibility. In the brain, neuromodulators introduce this degeneracy. They let the same basic circuit express totally different functional states or reach the same output through countless combinations of synaptic weights and neuron excitabilities.
Which is so different from how we design AI today. AI is optimized, right? It's usually designed to be a single, highly efficient, but very brittle path.
Exactly. And that optimized brittle system has a single point of failure. If that one connection breaks, the whole task fails.
Whereas degeneracy gives you
Resilience. If one pathway is impaired, or if one group of neurons gets exhausted, others can compensate to preserve the essential function. It ensures stability across different contexts, and it allows the system to rapidly reconfigure itself to achieve adaptability and find a new stable state based on what's happening right now. That's the flexibility we need in AI.
So to build this in, we need the right foundation. We can't use those abstract perceptron models from, 80 years ago?
No, they just don't have the biological fidelity. They can't support cell diversity or dynamic modulation.
Which brings us to spike-based neural networks, SNNs. The sources really position these as the ideal vehicle.
They are. SNNs are the bottom-up approach. They simulate the actual precise timing of neuronal spikes, which captures so much more about the brain's dynamics than models that just track average firing rates.
The big challenge, though, and you can see this in Figure 1 from the sources, is that classic engineering trade-off. We need models that are bio-inspired enough to work, but still scalable and, energy efficient.
Right. And that's where we need models that can really handle neuronal heterogeneity, the idea that not all neurons behave in the same way.
And the sources highlight 2 SNN models that really stand out here.
The first one is the Izhikevich model, or the IZ model. It's incredibly powerful. It's based on bifurcation theory, and remarkably, with just a few parameters, it can simulate the firing patterns of pretty much every known type of cortical neuron.
Like what kinds?
Things like fast spiking inhibitory neurons, regular spiking excitatory cells, bursting neurons, all of them. This makes it fantastic for simulating large, functionally diverse neural populations.
But there's always a but. If we're talking about efficiency, for robotics, say, doesn't the IZ model hit a computational wall?
It does. It has a high computational cost. The equations have these quadratic terms, which means while they are super accurate biologically, they demand a lot of processing power.
And if you're building a robot on a battery...
That's a non-starter. Those quadratic terms might be fine for a supercomputer doing research, but they could drain the battery on a small mobile robot in, minutes.
So we need a high-fidelity but resource-efficient alternative.
Which brings us to the generalized leaky integrate and fire neuron, or GLIF.
Okay.
The GLIF neuron is a really clever compromise. It enhances the basic, very efficient LIF model by keeping the equations linear, so no costly quadratic terms. Yeah. But it adds specific nonlinear rules for what happens after a spike.
And that lets it.
It lets it reproduce the huge variety of spiking behaviors you see in big databases like the Allen Cell Types database. So it's a really compelling option for anyone designing neuromorphic hardware who needs that biofidelity without killing their power budget.
Okay, so that covers the functional diversity, the different firing patterns, but the brain also has this incredible morphological diversity. We have to talk about dendritic compartmental dynamics.
Absolutely. The complex branching structures of dendrites are not just passive wires. They are sophisticated little computational units on their own.
And modeling that is complex.
It usually requires really complex multi-compartmental models, but we're seeing new efficient frameworks like Dendrify emerge.
So why bother with all that complexity? Why not just stick to the simpler point neuron models like IZ or GLIF?
Because dendritic trees dramatically improve resource efficiency. They let a smaller number of neurons handle far more complex computations. The classic example is the XOR problem. In an abstract ANN, you usually need at least 2 layers, multiple neurons, to solve it. But if you have a single neuron that correctly models the dynamics of a 2-compartment dendrite,
just one neuron,
that one neuron can solve the problem instantly. Dendrites move the computational power from the network architecture down into the cell itself. It saves energy and complexity.
So now we have these diverse digital neurons, We need to talk about how they learn.
Right. And we know backpropagation works well for isolated tasks, but for lifelong learning, you have to add these heavy-handed regularization techniques like elastic weight consolidation or EWC.
Which acts like a biological brake.
That's a great way to put it. It selectively slows down the updates to weights that are deemed important for tasks it's already learned.
But the real versatility, the real biological trick, comes when we don't just put the brakes on, but we actively link the learning rule itself to the context.
And that brings us to modulated synaptic plasticity and what are called neo-Hebbian methods. This is where we start building a true brain-inspired controller.
So you take a plasticity algorithm like STDP.
Spike timing dependent plasticity, yeah, which adjusts synapses based on the relative timing of spikes. That becomes exponentially more powerful when you integrate it with these global modulatory signals.
And the sources point to reward-modulated STDP, or RSTDP, as a key example. This is critical because it starts to solve one of the biggest challenges in AI.
The credit assignment problem.
Exactly.
Which, in simple terms, is the AI trying to figure out which action it took 5 seconds ago or even 5 minutes ago actually led to the success or failure it's seeing right now.
Which is really hard to trace back.
It is. But RSTDP helps solve this by acting like a chemical signal that carries that global information.
How does that work mechanistically?
RSTDP adjusts the plasticity window of the synapse based on a global reward signal, basically mimicking what dopamine does. Right. If a big reward comes in, the plasticity window opens wide to strengthen the synapses that contributed. If the reward is low, the window closes or even reverses to weaken them.
So it links the local learning rule, STDP, to the global outcome.
Exactly. It's a biologically plausible way to solve that temporal credit assignment problem.
And if we look closer at these chemical controls, it seems like each major neuromodulator has a highly specialized job in reinforcement learning. There are like specific dials on a control panel.
They absolutely are. Dopamine, DA, is the classic one. It's linked to reward prediction error, the better than expected or worse than expected signal that drives learning.
Okay, what about the others?
Serotonin, or 5-HT, steps back a bit. It regulates the balance between immediate and long-term rewards. It basically influences patience.
And then there's noradrenaline, NA, which is associated with arousal and vigilance.
Right. And in a learning context, noradrenaline manages the critical exploration versus exploitation balance. High NA, the agent explores new strategies. Low NA, it exploits what it knows works.
And the last one, acetylcholine.
SAGE, yeah. That influences the learning rate itself. It often signals attention or salient stimuli, telling the network how quickly it should change its internal rules.
This all leads to this proposed framework in the research, which tries to bridge the gap between these tiny cellular level effects and the overall circuit behavior.
And the goal here is to create SNNs with built-in neuronal diversity using IZ or GLIF models, where the neuromodulators act in a neuron-specific manner.
That's the key, isn't it? The specificity. You can't just flood the whole network with one signal.
No, you need cell-type specificity. We know from theoretical studies that heterogeneity matters immensely. For instance, having diversity in your inhibitory interneurons is vital for gating and directing how signals propagate through the network.
So if you don't have that cell-specific action, aren't you just swapping one artificial mechanism for another? Why is this better than the current AI regularization techniques?
Because it's dynamic and it's context-driven. Instead of a hard-coded rule like EWC that permanently penalizes changes, the biological framework uses cell-type specific neuromodulation to dynamically regulate the SNN's population threshold.
So you can, what, just make certain neurons less likely to fire?
Exactly. By slightly shifting the excitability of specific inhibitory cell types, for example, you can stabilize old memories without completely locking down the entire network. It fights catastrophic forgetting in a way that's plausible and sensitive to the context.
And these modulators are rarely acting alone.
That's really the final frontier. Dopamine's effect is incredibly complex. It depends on whether it binds to a D1 receptor, which is often excitatory, or a D2 receptor, which is often inhibitory.
So it changes the balance between learning from good outcomes versus bad ones.
Precisely. And the synergy is profound. The co-release of acetylcholine and dopamine doesn't just add their effects together. It creates a whole new, sophisticated control system that governs attention, learning, and memory all at once.
So why does all this complexity, cell diversity, chemical messaging, why does it matter outside of a research lab? Why should a robotics engineer care that their chip has to deal with quadratic terms or different neuron types?
Resource efficiency, it always comes back to that. Biological organisms are masters of optimization. They run these incredibly complex brains on the caloric equivalent of a dim light bulb.
Right.
Robots, especially edge devices, are battery constrained. They need robust online learning. They need homeostatic plasticity to handle perturbations in the real world.
Like what?
A robot that suddenly drives from pavement onto sand or has a joint that's starting to degrade. It has to adapt its motor control on the fly without being rebooted and retrained from scratch.
And neuromodulation gives them the tools to do that.
It provides the dynamic control to adjust synaptic strengths and excitability on the fly. It basically widens the robot's stable operating regime, making this framework perfect for motor control, spatial navigation, and true lifelong reinforcement learning.
But we always have to circle back to that trade-off. Modeling this level of biological detail is computationally expensive compared to a simple standard perceptron.
And that's where neuromorphic hardware becomes the ultimate accelerator. This whole SNN-based framework, with its sparse asynchronous spiking activity, is perfectly matched to neuromorphic devices.
Chips like Intel's Loihi.
Exactly. Those chips are designed specifically to exploit the low energy consumption that spiking computation offers.
And the dream here isn't just fast processing. It's having the chip learn autonomously, on the fly, right there on the robot, updating its world model the moment something changes.
That's the goal. And the fact that we've seen successful implementations of highly complex networks, like the entire Drosophila fruit fly connectome onto chips like Loihi 2, it shows that these bio-inspired models can be engineered for scalable, low-power adaptation.
Okay, so let's bring this all together.
I think the core conclusion here is that we have to move beyond this restrictive focus on just optimizing synaptic connections.
Right.
Incorporating the functional diversity of cell types and the context-sensitive control of cell-specific neuromodulation. That provides the essential degrees of freedom we need to build AI systems that are inherently robust, adaptable, and genuinely efficient.
Systems that finally start to mirror biological intelligence. So what does this all mean for the next decade of AI? It sounds like the future of intelligence won't be achieved just by making abstract networks bigger and deeper in isolation.
No, the breakthrough is in biofidelity.
In making them more accurate right down to the specific chemical messengers that let the network decide when to learn, how much to learn, and what to keep.
That's it.
So if the brain uses these neuromodulators as adaptive neural operators, providing this high-fidelity, context-aware control, what level of intelligence are we missing in today's AI? Just waiting for the right biological key, maybe a finely tuned blend of dopamine, acetylcholine, and serotonin to finally unlock true, lifelong biological intelligence. What could a machine with that kind of functional flexibility actually do?